Our next speaker will be presenting remotely. Oh, perfect. There he is. So I'd like to introduce Garth Covernton from the University of Victoria, joining us from my coast, West Coast, but of Canada. All right. Garth, can you hear us?
Yes, I can. Can you hear me?
Yes.
Okay. And can you see, can you see my slides?
Yes. Looks great.
Okay, excellent. Great. All right. So hello, everyone. Glad to be with you. It's kind of weird just talking into my computer and I can't actually see you. So I'm just assuming people are listening to me. But I am a Ph.D. candidate here at the University of Victoria in British Columbia, Canada. And today I'm going to talk to you a little bit about what we know about microplastics in seafood, including some research I've worked on myself, and then also a bit of a summary of where things are in the field with other folks' research as well. So, one sec. Why isn't my slide changing? Oh, there we go. Okay. So just some quick background on myself. For those who don't know me, most of my work has been on what we could consider seafood, although I'm generally interested in marine ecology and ecotoxicology. So I've worked on microplastics in bivalves. So these are the like clams and oysters in their habitats, and in their habitat in relation to shellfish aquaculture. I'll talk about a bit of that work. I've done some methodological work on sampling in seawater. There was a sort of study that made the rounds in the media on human consumption of microplastics I'll dive into. But currently, my big project over the last 2 years has been to look at microplastics in marine food webs. And so the food web dynamics of microplastics are really what I think about most in the day-to-day, and I'll get into those a bit too. But just to kind of frame what I want to talk about today, I want to kind of frame it by focusing on some commonly held beliefs. I've heard a few of them sort of at least implied today, and then also things that are very commonly talked about in the media. And I want to talk about maybe rethinking those a little bit and why we might think these things. So to start with, we often hear about seafood as being a huge source of microplastics for humans. Even if it isn't said outright, I think there's often sort of like an implicit assumption that perhaps it's the biggest source, especially for the average person. I've overheard people or been told stories of people in grocery stores saying, oh, I won't eat oysters because those contain microplastics and things like that. And you can see like I've just copied out a variety of different headlines from various organizations, like all pretty big organizations, talking about how seafood is a big issue for these microplastics. And so this is what people are being exposed to. The second one is how kind of relates to that first, with ocean animals being at the most risk themselves for microplastics. We often talk about plastics in the contents of marine pollution. I want to talk a little bit about, well, maybe why for macro that's true, but for the micro, we actually are not certain of that, and why maybe we should rethink that a little bit and think about it more globally. And then people often talk about microplastics as accumulating in food webs, which also sort of implies the magnification. So by magnification, for those toxicologists in the room, they'll know that refers to essentially the exponential increase in a contaminant as you go up through the trophic levels in a food web, meaning that like a top predator such as humans or whales would be at the most risk from these contaminants. So things like PCBs do this, and we know this and we can show it. But I want to talk about what we know about microplastics in this regard. So starting just overall with exposure via seafood for microplastics in humans, just a highlight of this study, probably some of you have seen this. So me and a colleague, and as well as with some other folks, reviewed 26 studies on various food, drinking water, and air in terms of the microplastic content as sort of just a preliminary analysis of what do we know about human exposure. And we used US recommended dietary guidelines for this. And so what we found was, based on what we know, we could calculate around 39 to 52,000 particles per year from food and drinking water. This is across the range of like sexes from childhood through adulthood. And then we have, when we combine that with inhalation, that number almost pretty much doubles to get to 74 to 122,000 per year. But we want to really highlight, my point here is that this only accounts for 15% of recommended caloric intake. So all of these numbers are have large ranges in uncertainty. And then also that is massively compounded by the fact that the majority of food items that people eat have not had microplastics measured in them. So that includes red meat, poultry, dairy, grains, fruits, and vegetables. I actually saw the first study I've seen on dairy come out just a few days ago, even showing like the order of a few particles per liter of milk, which was interesting to see. But to kind of highlight here, like you can see seafood here does come out as one of the higher sources, but the range is actually within sort of what we might be exposed to from bottled water and perhaps even added sugars. And with air actually probably exposing us to far more particles. And remember also, even when we're inhaling things, the lung clearance mechanism often for inhaled particulate is to move it up into the like esophagus, right? And then it'll be ingested. So actually inhaled particles can actually be a factor in ingestion. And I think that often gets overlooked. So just to highlight that there are a number of pathways into human food, and it's not always, and it's probably not at all about what an animal eats during its life, which is often the focus we talk about. So we already have only limited data on that, mostly in fish and shellfish, and almost none on these other food animals that we eat. But there's also a huge number of ways, right? So we live in these highly plastic environments. So, you know, our fleece jackets, our air filters, whatever, are shedding various microfibers and microplastics. Our carpets, right? We all probably have carpets in our homes that are made of polypropylene or rubber or some other synthetic polymers. And these are going to release particulate. And there have already been indoor studies showing like pretty high concentrations of these particles in our air. They can settle onto our food while we're making it during mealtime. There can be contamination during transport or sale if the product's open. During processing, we have no idea what actual, say, like a highly processed food, how that would compare to something that was, you know, packaged immediately, and how much that exposure to factory environment would affect microplastic contamination, and then also leaching from plastic packaging itself, right? So many of our foods come in these plastic wrappings. How does that affect microplastic content? We actually have almost no data on most of these things. Just as an example, to kind of put these things in a light, from some of my work here in British Columbia, we looked at clams and oysters on and off shellfish farms around BC, around Vancouver Island. And we actually found that there was less than one microplastic particle per shellfish on average, with most of the sources seeming to be these like textile fibers, which kind of leads us to the conclusion that, you know, and of course this varies globally and depending on your shellfish consumption, right? But at least for folks eating these shellfish in BC, for indigenous groups, they're going to be eating a lot more, perhaps more at risk, but for a lot of other people, this is almost like a gourmet item. And so really the exposure to microplastics via shellfish, for at least in this area, is going to be quite low relative to perhaps some other sources. And to put this into context, like if we take the average consumption of Canadians of shellfish and say they were eating half each of these two species that we measured, we could calculate that to be 87 particles per person per year. Putting this into context, there was a study from Caterino et al. In the UK, and they found they estimated 123 particles per person per year, which they then, in European countries where they eat more shellfish, they extrapolated to become 4,600, so 4,600 particles per person per year in these more shellfish consuming countries. But they also literally just put out filters and looked at particle settling, so mostly these microfibers during mealtime and also during food prep. They basically were able to estimate that this, compared to that, you know, up to a few thousand per year, you were actually looking at tens of thousands, so 13.7 to 68.4 thousand particles per year per person, if you take this calculation of what would settle into your food while you're just preparing it and eating it, right? And so I think this will probably hold, unless you're talking about extremely contaminated shellfish. This is probably going to hold. And so this is why I think this conversation needs to get reframed a little bit in terms of to consider both what's coming in with the food and then what is also coming into it later when we're preparing it or in the factory, et cetera. And just to even sort of further the conversation on microplastics, especially from the standpoint of shellfish, there's often a tendency to talk about bivalve shellfish as just passively accepting whatever particulates in the environment into their bodies, right? And this is actually not true at all. They're actually pretty selective filter feeders. So Evan Ward has published on this a bunch, and so I'm just basically citing some of his work here, where he found that mussels and oysters actually reject and or egest microplastics. So they have two phases, actually. They can first reject particles in their pseudofeces. So a lot of these bigger particles, they won't even ingest. And then once they are ingested, they can also fast track them through their digestive system, or they can digest them regularly and then poop them out that way. And all of these they're going to have differential residence times along the different steps, depending on the size and the shape of the particles. So they're not just passively accepting particles and eating them and accumulating them. The retention actually increased with decreasing particle size. So kind of to highlight other things we've been talking about, these smaller particles that are so hard to measure could be the greatest risk for actually accumulating. But generally, for what we're mostly measuring in the environment, and so what a lot of the conversation about microplastics has been on, these like, say 100 micron plus sort of particles, these mussels and oysters were actually pretty good. So you can see these numbers are around maybe 50% of those particles were either not even ingested to begin with or quickly passed out of the digestive tract. So they've actually been proposed as being not so good for monitoring microplastics in the environment. So kind of my question is, we often talk about microplastics and then seafood. These things often tend to happen together. And so I want to talk a little bit about like perhaps why that is and what we can do moving forward. So there is a big bias in the literature towards studying marine species, mostly because most microplastic scientists have come out of a background of marine ecology or marine biology or environmental science with an aquatic sort of standpoint. So you can see these trends over time from this Actigan and Guven review showing how as they increase, you still see this big sort of predominant for marine studies, with terrestrial and freshwater studies just starting to kind of take off, but still representing like far less of the field of research than marine. And this also holds up for ingestion studies. So this Provencher et al. Paper in 2018 looked at ingestion studies, and of the couple hundred of studies they reviewed, they only found 8 that actually looked at the ingestion in freshwater. And so really we are always talking about these risks and ingestion in terms of the marine environment. And so this gets translated often into human exposure as well, even though we're really missing data on all the other fields. Not to say that we don't necessarily need to be doing this research, but I think it's important to always keep it in context. We also know very little about how the exposure to animals that could transfer these microplastics to us, how this varies across the marine, freshwater, and terrestrial systems. So if you think about a marine system, even if you have a sewage pipe or something releasing all these textiles going there, usually that's going to be like a higher current area and things tend to get dispersed relatively quickly, as opposed to, say, a freshwater stream or a lake where these there's a tendency these could potentially concentrate a lot more, right? It's just less like dilute. And so you could potentially in these systems have organisms that are exposed to much higher concentrations. And I've heard some sort of both published and anecdotally from folks things about some of the Great Lakes, for example, having incredibly high microplastic concentrations relative to marine environments, but little knowledge of how that relates to perhaps like freshwater seafood species in that regard. And then if you think about terrestrial standpoint, we have almost no knowledge on how much terrestrial animals are consuming microplastics. And we've heard earlier about how we'll spread sludge, which contain all these microfibers and things into terrestrial environments. And also thinking about the fact that most of the plastic production and use happens on land, and yet we have very little understanding of how these microplastics are dispersing onto land. And we know that they move through the air, so we could expect that they're probably depositing on land to some extent at least. And what about humans as a top predator? So we often hear we're at risk because we're eating high in the food chain. So we're going to get these microplastics that have accumulated in animals and move to us. But while they're definitely getting into an aquatic food web, so this graphic just kind of shows how at all the different levels of all the different trophic levels in a food web, we do have evidence that these different animals are all ingesting microplastics. We also have evidence that trophic transfer, so movement between levels in the food web, is definitely happening. We can show animals eating other animals that contain microplastics. However, when we actually look at, sorry, one sec, slide is not changing. Oh, there we go. So when we actually look at the data, so I'll start with this image here just on the right. This is a recent study in ecotoxicology and environmental safety from Welkinshaw et al. In the UK. And so they basically looked at microplastics in terms of gram. So this is number of particles per gram wet weight of an animal. And as you can see, there's actually pretty much an exponential decrease with increasing trophic level, right? And so ecotoxicologists refer to this as trophic dilution. And so this happens commonly for things like certain PAHs, for example, that are metabolized by animals. But in this case, I think this is probably more the question of animals are pretty good at pooping these out before they are eaten. And so even if you saw perhaps a linear increase with in terms of like number per individual, for example, this is probably not so much like accumulation or magnification, as we like to say it, so much as just kind of, there's always some chance of adjusting these things. If you're a larger animal, you're eating more, you're going to ingest more microplastics, whether directly or indirectly, but that doesn't necessarily mean they're actually accumulating or magnifying, right? And so I think we need to kind of like reference some ecotoxicology work to talk about these things a little more appropriately. On the left here, this is a study I'm kind of working on right now, and we tallied up microplastic levels in marine fish globally, and I'm planning to add freshwater species to this as well to kind of talk about this. And so we pulled out trophic level for all of these species using the online database FishBase, and we can see that overall, there's no real trend for the number of microplastics in gut, and this is in terms of the individual. So even in terms of individual, you can get higher concentrations. This is an exponential curve on the left here. You can get higher concentrations essentially across trophic levels, and there's no clear pattern for larger animals ingesting more, suggesting they're probably not actually accumulating or magnifying. However, caveat, here we're mostly talking about particles that are greater than 100 microns. This is due to, as we've talked about, essentially the methodological gap when you go below 1 micron and often tens of microns even. Mostly because we're using visual screening and the human eye, even through a microscope, just can't pick out these smaller particles. And if we can, there's a huge bias to them and we miss a lot. So we also know that these smaller particles tend to become exponentially more prevalent as they decrease in size. So while we know like animals may be better at removing these larger particles out of their gut at least, we have little data on what could be happening with these smaller ones, both in terms of the gut and with translocation into organs, liver, things like that, right? So these tinier micro and nanoplastics could still be accumulating and magnifying, but we have almost no data to suggest either way on this. We do know we have this nice study from Collard et al. In 2017 found little chunks of, it was an aggregate of of a plastic, I'm forgetting the exact polymer, but with cellulose, and they were able to find these chunks in liver cross sections that seem clearly to have somehow found their way into these anchovy livers. So we have some evidence that perhaps smaller particles can either directly move in or come in as a nano level and start to aggregate in organs. But this is, as far as I know, one of the only studies that really shows this in a way that's pretty undebatable. Yeah, and so that's kind of what I wanted to say today, just to talk about where the data gaps lie. I think that human ingestion of microplastics via seafood should not necessarily be ignored, but I think it needs to be put into context with other sources of exposure. We need to start doing research on all the other things that people are eating, as well as the drinking water and inhalation, as we've already talked about today. It's going to vary really depending on the species you're eating, and as the earlier speaker talked about, like where in the world you're living, what kind of seafood you're eating. So are you eating the whole gut of fish? And then also the proportion of your diet that is seafood. So if you're perhaps if you're an indigenous person or someone really dependent on subsistence living lifestyle, you are potentially eating a lot more seafood. So we need to think about those different ingestions as opposed to an average, and then also all the other routes. And especially I think things like plastic packaging and factory conditions need to be considered. And while we focus a lot on marine species, I think we need a lot more work on freshwater species as well as terrestrial animals, so like livestock and things that folks are eating. And as kind of said, the larger microplastics do not seem to be accumulating, but rather diluting in food webs. But we should think more about these smaller ones to try to understand them. And so that means kind of, to me at least, that while humans, we often think about us eating microplastics via our food and getting them from animals. But I think that our exposure is probably high not because we are eating all these food animals that are eating them necessarily, but more because we're already living in these highly plastic environments. We're in a high sort of particle load, microplastic load already, and it's not necessarily this food web accumulation that's the factor that's leading to our higher exposure. So with that, I'm happy to end and take any questions if I have time.
Wonderful. Thank you for that excellent talk. You had a room full of very attentive people, so no worries about speaking to an empty room at all. We do have just a few moments for questions. If anybody has any burning questions after this talk, feel free to hop up. Please go to the microphone, though, at the side of the room, just so that everybody on the can hear you online.
Can you hear me?
Yes.
Hi. Juliet Cohen with the Plastic Pollution Coalition. Thank you so much for this terrific presentation. I'm super curious about the chemicals, though, in the plastic and the difference between testing for just the actual micro or nanoplastics and actually also testing for the bioaccumulation potentially in ocean life and human of the chemicals in plastics. Are you looking at that too?
So that's not my focus of research per se. I'm hoping to do more of that in my future, but I haven't looked into that personally. I would actually defer almost that to Dr. Clemens, who's in the room, and he published a study in the last couple of years where they actually looked at the potential for accumulation of these plastic-associated chemicals and found that the environmental exposure is a lot more important, and then actually as you increase your microplastic load, these pollutant exposure actually goes down because microplastics are so readily able to absorb other contaminants. But like I said, that's a little more complicated than what I might know about.